Dengue is of special interest because nobody realizes the impact it has. There are about 400 million people estimated who get infected. At least 100 million people are symptomatic. The mortality could be as much as 1%. When you start looking at 400 million infections, that's a huge number.
Primarily, the infections are documented in the tropical countries.
There are no vaccines, there are no antivirals, there are no therapeutics. So the question is, how do you tackle such infectious diseases, especially in the developing world where the burden is the largest? It is to look at human immunity. And so you need to have in-house capacity to be able to capture the human immune response and to understand it. Our clinicians collect hundreds of blood samples for us across the spectrum. And then what we do is we get these blood samples and then we isolate the cells from there, we isolate the plasma, and then we ask critical questions that will help us better understand what is happening at the level of human immunity.
We are choosing the individuals on the basis of their high titers of dengue-specific neutralizing antibodies. Then we are collecting their bloods. In the lab, we are isolating the human monoclonal antibodies by amplifying their antibody genes and tested for their in vitro binding and neutralization assays against a particular pathogen. After the screening, we identify a cocktail of antibodies which has the capacity to neutralize all 4 dengue serotypes.
So that we can then use this information and use all this knowledge to better design vaccines. Human monoclonals can protect where vaccines cannot reach. You need them for people who don't respond well to vaccines. So the way my lab works is that we have the capacity to identify the cells that are making antibodies to the pathogen. Once we isolate those cells, we can clone the antibody they were making. And when we clone it, we put it back into tissue culture, make the protein, which is the antibody, which is a fully functional human monoclonal. Because while the world waits for vaccines, we need alternates. So the platform that we set up for the human monoclonal technology at ICGEB, we not only used it for dengue, we also used it for chikungunya. That's another viral infection that also is mosquito driven. And while it does not have very high mortality, morbidity is severe.
Our lab already has an expertise to develop therapeutic antibodies to dengue and chikungunya. And when COVID-19 emerged in 2020, we identified a very potent, highly broadly neutralizing antibodies to SARS-CoV-2, which is effective against all the Omicron variant. ICGEB stands for International Center for Genetic Engineering and Biotechnology.
It's a UN institute. It's in 3 different parts of the world. The entire vision of ICGEB is to work for the developing world and get biotechnology to everyone and anyone who is in need. And our lab goals perfectly align with SDG 3 of the Agenda 2030 of the United Nations, because we are looking for solutions in the form of vaccines and therapeutics to safeguard human health.
If we are successful in developing neutralizing antibodies to dengue and SARS-CoV-2, we can save so many lives. In future, if there is a newly emerged virus, the information gained from our ongoing research can be quickly utilized to provide immediate solutions to that particular pathogen.
And when we get all this together, I'm very hopeful that we will find solutions in the due course of time.